Good afternoon, and thank you for joining us. Michelle, thank you very much for the over generous introduction, and special thanks to Biogen for hosting this event and a good bit more. At Ionis, we and Biogen have been collaborators for many years in the neurological disease space. And that collaboration has been tremendously productive and a great deal of fun. In addition, Michelle and Biogen have been instrumental in the formation of n-Lorem and instrumental in all the progress that we've made. So we thank the wonderful people of Biogen and with deep appreciation. n-Lorem is a charitable foundation that I initiated in January of 2020. In the next few minutes, then, I'll introduce you to n-Lorem and summarize the exciting progress that we've made in our first year plus month or two. Our mission at n-Lorem is to take advantage of the technology that we created at Ionis to discover, develop, and provide experimental ASO treatments to patients with ultra-rare mutations, and to do all that for free for life. The n-Lorem patient, that is the patient with the ultra-rare mutation, presents a host of challenges that are as unique as the mutations themselves. It is absolutely true that in many cases, these mutations are so unique that there's a single patient in the world that has that specific mutation. Thus, each patient requires a specific medicine to be created for that patient and that patient alone. So it's a micro-scale problem. We've also learned that even within the larger genetic diseases that are well understood, there typically is a common mutation that causes the disease in most patients, and then a number of unique mutations that produce the same problems, but are not going to be solved with the medicine for the common mutation. Once again, the patient requires a specific medicine for that patient and that patient only. But it is the ultra-rare patient presents also a macro-scale problem. Thanks to the sequencing of many, many human genomes, we now know that there are millions of these kinds of patients. So any potential solution for treatment of any of these patients must be scalable. In addition, the diseases caused by ultra-rare mutations are typically very severe. Often they're rapidly progressive and can rapidly become fatal. And so these patients have an urgent need, and they have a desperate need. Moreover, most of these patients will have spent several years trying to learn what is the problem that's causing their health to be so poor. And that journey is both long and perilous. Lots of negative things happen to these people. So very often the diseases are advanced and adding to the urgency. And finally, these patients and their families face their problems alone. They're unique. There's a single patient and a single family that has to deal with this. So there is no community. There is no set of patient support groups. And there are no physicians who've treated 10 or 20 or 100 of such patients. And I think that isolation results in even greater damage to the family than you might see with a rare disease. And today, n-Lorem is the single institution that's offering immediate hope and rapid treatment for many of these patients. So to put a face on the words I just told you, let me introduce you to Connor Dalby and his family. Connor is a patient we're about to begin treating. And I think his story is very much a story that we hear every day.
I'm Kelly Davey. I'm a mom of 3 boys, Chase, Connor, and Cameron. And one of the most difficult things for me to balance as a mom to 3 boys is I have 2 kids that are completely healthy and typical in every way. And then I have Connor, who has a lot of special needs and needs constant attention. When Connor was born at a few weeks old, we noticed something was a little bit off with the way he was eating, crying often. He wasn't sleeping. He was very uncomfortable the majority of the time. When he was 8 months old, he started having seizures, and we started trying a lot of different anti-epileptic drugs that are available. And nothing was really helping to reduce the seizure frequency. And he was averaging about 50 seizures a day. So all day, all night, he was having seizures.
The Davey family story is sadly very typical. Connor was born with a unique mutation, and the family spent years trying to understand what was wrong with Connor.
He ended up being placed on hospice services when he was 3 years old, and they didn't think that he would live much longer based on the severity and frequency of his seizures. They said he had a mutation in what was called the SCN2A gene. Very little was known about that. They had nothing available to change his treatment or help him. It could end up being very severe, or he could improve, and they just didn't know.
The mutation caused severe seizures, developmental delays, movement disorders that were difficult to control.
What we found was that Connor's mutation in the SCN2A gene was incredibly unique. And basically, he would need his own sort of treatment just for him because his gene mutation was not like the majority of the other kids with SCN2A. And so for me, it was about that time that I started really losing hope because I didn't know what else to do to help him. My son's now 11 years old, and the seizures, they still get me every time to watch my child have a seizure, and there's nothing I can do to stop it. It still breaks my heart, every single one, and I've seen thousands, and it never really gets easier.
Eventually, they learned the cause, only to learn that Connor is unique. There's not a single other person in the world who has his mutation. Only through n-Lorem is there hope for Connor.
I feel like I've been fighting for so many years alone with my son, for my son, and I wasn't very successful in helping him. And n-Lorem, what is happening right now with a team of scientists and doctors and donors, people stepping in and giving their time and their energy and making a medication for one child, for my son, to help him and his quality of life and our family. It's still so hard for me to believe and accept that all of these people are coming together to help Connor.
We are blazing new ground. My greatest concern is that because we don't have sufficient financial resources someday, I'm going to have to tell a patient we could help that we can't. So these stories are desperate and tragic, and it's very clear that something needs to be done. And n-Lorem is attempting to do its part in creating solutions for these patients. To even contemplate doing what n-Lorem is doing, my feeling is that 4 components have to be in place. Of course, the first step is to have a technology for drug discovery and development that is efficient enough, versatile enough, and broadly enabling enough that you could even think about doing this. The second task for me was to determine if we could somehow use the commercial model to meet the needs of these patients. And there's a lot to recommend the commercial model because we know it, we understand it, we know how to use it, and it is sustainable. But I rapidly concluded that it simply wasn't possible using a commercial model. And so we are creating a charitable solution. We are blazing new ground. Every step we take is a step into the unknown, and there is no map. The map that we are creating at n-Lorem will become the map for how to do this. The third element is that we must have access to patients who are genetically appropriately characterized and are under the care of a physician who's a research investigator capable of taking care of this patient under an experimental IND. Fortunately, I was introduced to the Undiagnosed Disease Network. The UDN is a consortium of major medical centers funded by the NIH who've made it their goal to have the opportunity to diagnose patients who have been undiagnosed and to characterize them genetically. And so I developed a collaboration with the UDN very early in the development of n-Lorem. And that collaboration certainly has been instrumental in the progress that we've made. But interestingly, less than half of the applications for treatment that we've received have come from UDN institutions, and that's great news. That means many other institutions recognize that this desperately needy patient population is underserved. Actually, not underserved, not served at all. And many people are turning to, and of course, we need all of them. And then the final element that I felt had to be in place is that the regulatory approach had to be supportive. So early in 2019, I introduced the idea of n-Lorem to the senior leaders of the drug division at the FDA and policy people at that division. And we are supervised by the drug division. And was very, very thrilled with the response I got. It was very rapid and very supportive. I was even happier than when the FDA announced that it was going to develop guidance for the treatment of N-of-1 patients. And in the development of guidance, there are always a public comment phase. And so we took the opportunity to make two very substantial proposals to the FDA that laid out very specifically the paths that we think are optimal to meet the needs of these patients. I was even further pleased then in January of this year when the FDA published initial administrative guidance. And what pleased me about it was that the guidance was focused on ASO specifically and ultra-rare patients, and that it emphasized that all stakeholders involved in this endeavor have to be collaborating and to support these patients. And then finally, the FDA has indicated that it will provide definitive guidance later this year or early next year. And we look forward to that. Realistically, we don't expect it to have the FDA to agree to everything that we suggested, but we do think that we're going to have guidance that supports our doing the work that we need to do. And we had the opportunity in parallel with establishing n-Lorem to help two investigators actually obtain experimental ASOs and administer them to now 14 patients. And that's Tim Yu at Boston Children and Neil Schneider at Columbia. And that was really important experience for us because it gave us a real life opportunity to deal with the FDA with these specific type patients. And we're quite encouraged that even without formal guidance, we are able to work with the FDA and get treatments to these patients in time to help them. So we thank Neil and Tim for that opportunity. And so I think we can be very confident in saying that n-Lorem is already working. The n-Lorem model is working. The simplest way to think of n-Lorem is that we're a bridge between the need, which is the appropriately characterized patient, and a research investigator capable of taking care of that patient, and the opportunity for treatment, which is the technology and drug discovery and development engine that we build at Ionis. Specifically, n-Lorem provides a site to which patients or parents and research investigators can apply for treatment. If we determine that the patient is appropriate to be treated with an ASO, then we fund Ionis and various contract research organizations to discover, develop, manufacture, and provide the ASO to the patient for life for free. And then during treatment, we support the patient and the physician, and in particular during the clinical trial phase of the treatment, with substantial effort to assure that we learn the maximum amount from each patient. I want to return to the technology for just a minute. And I'm going to be making some broad assertions about the technology, and I don't have time to share the data to support those with you. But my colleagues and I have written quite a number of reviews in the last year. And so if you're interested in learning more about the technology that makes this possible, you can certainly begin there. The intrinsic efficiency of Antisense technology was no surprise to me. It was one of the major reasons that I founded Ionis to pursue it. But that alone would not be sufficient. A key component of what makes this possible is that today's Antisense technology is quite versatile. We know how to design ASOs so that we can take advantage of multiple post-RNA binding mechanisms, and that increases the breadth of utility of the technology. We validated that these medicines can be administered by all routes of administration. And we know that when we administer them systemically, they distribute broadly, and we understand their behavior in the major organs in great detail. In such detail that I can tell you how many molecules per cell in various organs we need to see a pharmacologic effect. The Ionis automation is also critical and unique to Ionis. It's a product of 30 years of experience and then a great deal of AI learning. And that then simplifies and makes very rapid and very inexpensive the identification of the site in the RNA that would give us the best ASO. We typically will screen more than 1,000 sites in a target RNA with multiple designs of ASOs to identify the very best ASOs. That process is highly efficient and very low cost. We simply add a few plates to the robot at night. Another key component is that within a specific chemical class, all the medicines are basically the same. They just differ in their genetic zip code. So we know then how the next drug is going to behave because we have experience with another 20 that preceded it. So that's very important because we have a very good idea of what dose to use. We know how frequently that we have to dose. And because we have constructed databases that integrate all safety observations from non-human primates through all controlled clinical trials for all the chemical classes that we have in development and commercially, and published those and provided them to regulatory agencies, we have a very good sense of what to watch for. And I think that the fact that those databases exist and are available to regulatory agencies is an important component of their positive response. Finally, just the potency and duration of effect of modern ASOs means that the cost of treatment is much lower. I mentioned that we have multiple post-RNA binding mechanisms that are available to us. This slide, it just shows a bunch of cartoons of different approaches we can use to cause either a reduction of the target RNA and the protein or specific reduction of the protein. More importantly, in the last few years, we've learned how to use newly discovered regulatory elements in messenger RNAs to actually increase the level of the messenger RNA and the translation of the protein. And that means that we can work as an agonist as well, that is, replace some gene functions. And so all of these mechanisms are available to us, and more are coming all the time. These are the organs on which we're focused. Why these organs? We understand them. We have a lot of experience in them. We know how to work well in these organs, and our medicines are extremely potent. And that, of course, increases the safety margin. So, and we have 2 organs, liver and kidney, that we approach systemically, subcutaneous administration, and then 3 organs that we approach locally, which further enhances safety. the central nervous system, which we inject locally into the cerebral spinal fluid, the lung by aerosol, and the eye by direct injection in the eye. And of course, as we mature the technology and our understanding of it in the coming years, we hope to add other organs as well. So how have we done in our first year? We're thrilled. We think the response to n-Lorem has been extraordinarily positive. And that reception is being manifested in many ways. Importantly, we've established quality systems that I'll come back to and discuss in some detail. One of the things to remember is that we're providing very minimal preclinical information before we treat these patients. And that means that quality at every step even matters more than normally. I'd not intended to seek new donations in 2020 because I wanted to prove that this concept could work. Despite that, we've added somewhere between 25 and 30 new donors and $10 million in donations. And that encourages me to believe that we will be able to raise the funds to scale the technology to meet the need. n-Lorem can't do this alone. That's very clear. The only way this can be done and done efficiently, so we help as many patients as possible, is to establish a network of collaborations of all organizations interested in serving these patients. And we've had great success. All of our contract research organizations that perform animal toxicology studies for us are contributors and also partners and provide these studies at dramatically reduced costs. We've also contract manufacturers who are donors and provide production of these medicines at the lowest possible cost. Just in a year, these collaborations have allowed us to reduce the cost per patient by more than 40%. And that means we can treat more patients per dollar. Because of COVID-19 and because the novelty of n-Lorem, I thought we might see 4 applications in our first year. We're now approaching 80 applications received, and we're able to treat about a third of those patients. I do expect that third to go down a bit as the range of applications diversifies, because there are some things we can't fix. For example, we cannot fix a mutation that causes total loss of the RNA or the protein, a null mutation. We're very excited that in addition to those 14 patients with whom we have experience, we expect to be treating our first 5 to 7 patients under the n-Lorem umbrella in 2021, and perhaps more like 20 new patients in 2022. And of course, we have to get the word out. And awareness of n-Lorem continues to broaden. I'm told I made more than 200 presentations last year on n-Lorem, and I'm planning to make about the same number this year. And we really just have scratched the surface. The more people, the more physicians, the more patient advocacy groups know about n-Lorem, the more patients we can help. Quality. The first step in the process is these very complex risk-benefit decisions about whether a patient should be treated or not. Will an ASO make that patient meaningfully better? These patients typically are desperately ill. They have many signs, many symptoms, so they're very complex. And the process that we use is we have established a committee we call the Access to Treatment Committee that's comprised of all the expertise that we think we need to make these complex judgments and be confident that when we treat, there's a reasonable probability that we'll be able to really help that patient in a way they need. The next step is the development of a treatment plan that's specific for each patient. Every single patient gets that patient's treatment plan. We do that once we decide to treat the patient by working with the investigator. to define a primary treatment goal, secondary treatment goals, and exploratory goals, and very clear clinical measures that are validated that we will use to measure these. That way, we know what we're treating, we know why we're treating it, we're confident that if we improve that, that will make a meaningful difference in the patient and the family's lives. And it sets us up then to evaluate the performance of that ASO in that patient, because it takes about 10 months to a year between decision to treat and when we're ready to treat, just to do all the discovery and development work. And so we ask that the patient or parent and physician focus that year on collecting detailed natural history data with regard to all of those treatment goals, using the clinical measures that we're going to use, and to do that within the bounds of good medical treatment. And so once we begin treating, we have a very detailed, immediate natural history that's just very brand new. And then we'll be able to compare that to how the patient behaves during the treatment phase. We work with Ionis that assures that the very best ASOs and the highest quality ASOs are provided to these patients, and they deserve the best. As I mentioned, we've already established many important collaborations. Ionis, of course, is our most important collaboration. Biogen, a founding donor and collaborator, vital. The UDN, extraordinarily important. And all of these organizations have joined us. And we think we're just beginning. There are many biotechnology and pharmaceutical companies that we look forward to having us join. Patient advocacy groups of various sorts we are chatting with. We know that we can't do this alone, that we need the support of everybody who cares about these patients, all the organizations and individuals and all the patient advocacy groups. We need your help. And the door is wide open to become donors and collaborators and supporters. And something I've learned in doing these collaborations is that doing them is very good for the companies who join us. Every single organization we've collaborated with tells us that working with n-Lorem is a tremendous positive for worker commitment and morale. And of course, n-Lorem presents the industry and the companies involved with us in the best possible light. And it displays the true heart of our industry and the true heart of the people who come to work every day in the companies who are collaborators with them. We think that's great value. So learning from each patient. I think it is vital to maximize what we learn from each patient. We owe it to these patients. And they're incredibly important opportunities. For example, when there's a treatment, that's a great stimulus for patients and physicians to work together to identify more patients that could be treated. And so we're very quickly learning what the true prevalence for each of these N-of-1 mutations may be. As I mentioned, we'll have the opportunity to learn the natural history in all of these different mutations and all these different patients in some detail. With the ASO, and as specific as it is, we have the opportunity to prove in the human being that that mutation of that gene is the specific single cause of all of their problems. And because we have patients of all ranges, age ranges, and many genetic disorders begin shortly after birth, we are going to have the great opportunity to determine how much plasticity each of our organs has, especially the central nervous system. Many of our patients have genetic diseases of the central nervous system. And those genetic diseases cause then almost always developmental delays, delays in developing intellect, motor skills, and the like. And so as we treat, we're going to have the opportunity to see what, if any, of those developmental losses can be recovered if you take care of the primary cause of the problem. And finally, from an Antisense specific perspective, we're going to take advantage of the opportunity to evaluate new mechanisms. Many of the new mechanisms that we have that raise the protein levels use elements in the RNA that are not conserved from species to species, and that makes drug development very difficult. And here we have the opportunity to evaluate those mechanisms in the ultimate test species, man. So how are we going to do it? Well, I've mentioned that we will have predefined treatment plans and then well-characterized one-year natural history studies. We will treat the first year as a clinical trial. And so during that one-year period, we'll be collecting lots of data, and we'll analyze each patient in the aggregate experience that we have and publish. After that 1 year, then we'll just monitor the patient and, of course, have safety reporting and annual reporting. We're encouraging all of our investigators to publish their experiences. We plan annual investigator meetings so we can all share insights into how to do this better. We'll publish our aggregate results annually, and we are also developing a patient support system unique to n-Lorem. We think all these things are critical to both take care of the patient in the best possible way and to learn the most and share what we learn with the largest scientific and medical and patient communities possible. I thought I'd share with you a few of the patients we're moving toward treating now. And over on the left, you can see it's a very diverse range of genes already, a diverse range of investigators and institutions, some members of the UDN, others not, a very wide age range of patients, multiple mechanisms of ASOs, including allele selective agents. You can see there's a heavy predominance of central nervous system disease applications, but we now are getting eye and lung and liver and so on, and kidney. And so we do expect that the number of organs to diversify, and then that will add a lot more learning and a bit more complexity to our challenges. So what's next? Well, we're working with the FDA to assure that the guidance that the FDA issues is appropriate. It's sufficient that we can meet the needs of these patients and that we do it with the highest possible quality. We're excited to be initiating treatment of true n-Lorem patients this year. As I said, we'll be adding many more kidney, lung, liver, and eye cases. We look forward to publishing our annual evaluation of our performance. We're just completing materials to get out and formally fundraise. And so you may have us knock on your door one of these days. And if we do, I hope you open it for us and think about us as one of the places where you might consider putting your charitable dollars. We continue to add new collaborations almost weekly. We've barely scratched the surface. There are hundreds of biotech and pharma partners that we are confident will someday be collaborators and donors and supporters. We're especially anxious to make sure that all the patient advocacy groups who are relevant know we exist so that more patients can be referred to us earlier. And because the demand has greatly outstripped our expectations, we're moving from what basically is an all-volunteer staff now to some workers who we pay. And we need your help. We cannot do this alone. We cannot do this alone. This is going to take the best efforts of everyone who cares about another human being who's suffering and desperate. And you can help. All of you have a large interactome, to use the modern scientific term. You know a lot of people. Think about n-Lorem. Spread the word. Maybe I'll have to only make 199 presentations this year. We look forward to collaborations, both with patient advocacy groups, other biotech companies, pharma partners, and if companies who have other technologies feel at some point that they're mature enough to join in this, we'd love to collaborate with them or make them a part of n-Lorem. And we need you to consider being a donor. Every dollar, every dollar, every dollar matters to these patients. So I'll conclude by saying, I don't have a big dream for n-Lorem. It's a tiny dream. And that is that we'll change the future of one patient and one family at a time. And we'll change it for the better. And while that is tiny, I think in the aggregate, that very much is a wonderful way to change the world as well.